Alright, in this video I'm going to cover the very basic steps of DNA replication. The first step of DNA replication is that we have to open up the DNA, right? How will our enzymes who don't have eyes and aren't reading DNA like we do, how will they read it if it is not open and available to them? So we need to unzip the DNA, and that forms what we call the replication fork. So you can see here that the two strands sort of split, sort of like a fork in the road, okay? So we call that the replication fork. In fact, this whole area would probably be considered the replication fork. And the enzyme helicase, as we know, all enzymes end in ase, and this helic prefix, or sort of beginning here, reminds us of the DNA double helix. And helicase is an enzyme that will unzip the DNA by breaking the hydrogen bonds between the bases. And so here, this green molecule is going to be helicase, and it is right there at the beginning of the replication fork because that is the enzyme breaking the double bonds and basically creating the replication fork. And it will move down the DNA strand, continuing to open up the DNA as it goes. After the DNA is open, another enzyme, DNA polymerase, adds new nucleotides to form a new strand of DNA against the template strand. So just like we did in the other video, if there's an A, DNA polymerase will attach a T. If there's a C, DNA polymerase will attach a G. And let's just break that word down, just like you know that I do. ACE obviously means it's an enzyme. And then look at this, a DNA polymer, right? This is an enzyme that makes a polymer of DNA, and it's an enzyme, okay? And so this makes sense. We're adding nucleotides together, forming a polymer, a new strand of DNA against the template strand. So in this picture, we see helicases. In this case, they show two at the replication fork. And then we see DNA polymerase following helicase down the strand. So as helicase opens the DNA, DNA polymerase follows and adds a new strand. And so you can see here, the blue is the template strand. That's the dark blue. And the light blue is our new strand that DNA polymerase is creating. And so looking at the big picture here, you would think that seeing as our chromosomes are linear, right, they form lines, you would think, oh, why not open it from one end like you do the zipper of your jacket? Well, you could do that, but instead, we actually open DNA from somewhere in the middle, okay, at what we call origins of replication, and opening it in the middle forms what we call a replication bubble. And therefore, we have helicases at the two different ends and replication forks at the two different ends, moving in opposite directions. And this allows us to copy DNA twice as fast. And in fact, we could possibly have multiple replication bubbles going on a single strand. And as the replication bubbles continue, they get larger and larger, as seen in this picture, until they merge together, and we ultimately end up getting two double helices, each made of one old and one new strand. And of course, our end result is two identical copies of our chromosome, which will become sister chromatids when we study cell division.